So with you know, prokaryotes specifically, because they have small genome, and also with viruses, right, which we'll see as we go forward, they're able to switch around their genome to then meet environmental challenges. So like I was saying, for example, if a bacteria colony that is most viable at, let's say, a pH 7, and then all of a sudden you change that pH to a pH 8. Doesn't seem like a big change, but it can wipe out that colony. But what can happen, and we'll see as we go towards the end of this lecture or this presentation, the bacteria can switch up, keep changing its genome to then come across a version of itself, a mutated version of itself, in which it can survive in the pH 8. And now those bacteria will survive. And that's how we go on to create these bacteria that become antibiotic resistant. Okay? And then, of course, we'll see as we go forward. So this is just a small illustration to explain that when there is a challenge that challenges a certain large percentage of the bacterial colony, the bacteria are going to switch on this mutation, and then they'll go through all the different versions that they can until they come across a version or a mutation or strain that can withstand that environmental assault. So when we talk about genetics, we're talking about genes, and we'll see what genes are. But genes are basically all of our genetic information, right? Everything that we are is coded in our genes. So oftentimes I explain genes as, you know, when you have a cookbook, right? And what do cookbooks have? Cookbooks have recipes. And what do recipes produce? They produce their product, whatever it is. Let's say if it's you know, cakes or what have you. Sarah's got me thinking about baking now. So so you have cakes or breads or what have you, right? There are whole different kinds of cakes and all different kinds of breads, and each one has its own specific recipe. So similarly, our genome consists of genes, and our genes then have the instructions to make certain proteins and to make, well, not well, our genes are the recipes to make each one of our cellular products, so for proteins and so forth. And so that's why when you see identical twins here, they share the same genome. So that's why they're both of these you know, people that have identical genomes are going to look the same, right? They're going to look the same because they have this same, it's the same recipe book, right? So same recipe book is going to produce the same recipes and therefore the same products. But just to know that it's really not that simple, you know, specifically for humans. So this is a good picture because while they look alike a lot, notice that, you know, she's got this that's a little bit different and she doesn't. So that's how our genomes work also, because you can have the same genetics, but the expression of our genes, or the expression of the products of our genes, also is influenced by environmental factors. Okay, so especially, so for instance, in some diseases that have, in you see some diseases that, even though you're looking at a set of identical twins, it's often where only one twin will develop the disease and the other twin doesn't. So that tells you that getting diseases isn't always, you know, based on genetics. So schizophrenia, although schizophrenia is a umbrella term, but there are aspects, there are different diseases within that umbrella term of schizophrenia that you don't see in both the twins, right? So that's telling you that this is not an only a genetic reason. Okay, so in this case, in this picture over here, what they're showing is an E. coli, and in this E. coli can acquire. So this E. coli could be not pathogenic, not virulent, but if it conjugates with another, if with another E. coli that is virulent, then it can become virulent. And we'll talk about that transformation and so forth going forward. So prokaryotes can acquire pathogenicity. They can acquire virulence, okay? And that's how virulence and pathogens actually thrive. So genetics then is the science of heredity, of genes and how genes are inherited and what they code for and so forth. Heredity, or the genetics, is then the central dogma of molecular bio. Okay. And this central dogma of molecular bio is, we'll see coming forward, but the steps of the central dogmas of molecular bio is what leads to the explaining or explanations of mutations, and then how gene expressions can be controlled by factors outside of that gene. So you have your recipe, but I'm going to control the making or execution of that recipe outside of the recipe. Meaning, inside of the recipe would be, cake, flour and eggs and what have you. But what if I control whether or not you can turn the oven on? So you can't turn the oven on. You can have all of this mixed and ready to go, but you can't make that product, right? So that's what this means. Gene expression controlled by operons. Operons means operators outside of that gene. So the central dogma of molecular bio, which is the study of genetics, is that information will always go in this direction. Information will always start off from DNA. Parts of the DNA, specifically called genes, are going to have a copy made of them. That copy is then called an mRNA. That mRNA then leaves the nucleus. If we're talking about eukaryote, if it's prokaryote, there's no nucleus. That mRNA is then is the instructions, right? And then the instructions are going to be used to make the product from that mRNA, which is mostly a protein. And then the protein is going to get folded in specific ways, the way it needs to, within the endoplasmic reticulum, within the Golgi apparatus, depending on what that protein is. And then you will have its function. So how can a mutation, so what happens when you alter a genome? You end up with a mutation. So what does that mean? That means that this DNA can get mutated, a section of it or all of it. If you now have a mutated gene, then you're going to have obviously a mutated RNA. You'll have an altered protein, and that altered protein will have an altered function. Now, this altered function could be beneficial or it could be detrimental, depending the change and what it does. So mutations aren't always bad; they can also be good, right? So I'll talk more about this mutation after we've actually learned what ATGC is, right? But this is how this mutation occurs. So what they're showing is this. So in this case, you should have had, so notice the TAA now. So TA, you should have had a C in that place, but except now there's been an issue and that C has been replaced by this A. So that then translates to a mutation. So this is called a base substitution. When a base, a nucleotide is substituted, you can have where there's a movement or a deletion. There is no substitution. So in this case, we're back to our TAC, and now the C is missing, right? So then its partner will be missing also. So this becomes an issue. Okay? So this will make more sense once we learn what these pairings are. We'll talk about inducible and repressible operons after we learn about operons. Okay. So these alterations in these genes, as I said earlier with our mutation, can lead to a mutated mRNA and then mutated product, right? So alterations in bacterial genes and gene expressions can be the cause of diseases, can even prevent disease treatment because now it's a different microbe or a different organism. And this can be manipulated for our benefit, but it can also harm us, right? If you want to think about the flu virus, right? We have to get different flu vaccinations almost every year is because the flu virus mutates. So whatever antibodies that we've come up with, not antibodies, whatever antiviral chemicals we have won't recognize the new strain. Okay. And we'll talk more about that when we get to the different antibiotics, right? So we will also come back to this after we've gone through this chapter. Freezes. So we just go through some terminology. So genetics is then the study of genes, how this information is carried from the genes, how the mRNA is made, how is that information expressed and then how do those genes then get replicated? So in other words, you have the gene, the recipe that's in the book of DNA, in that recipe book. Well, if I want to make that recipe, so if the book is in the store or is in the bookcase, but I want to read a certain recipe, well, I need to. So if, in other words, if the book is, I'm pointing like this because there's a bookcase behind me, but I forgot to have that virtual screen on. Okay, so if you have a bookcase and you have that recipe book in the bookcase, now if I tell you you have to make this recipe, so you have to go look up that recipe in that book, but you can't remove the book from the bookcase. So that would mean you'd have to make a copy of that recipe, and then you can only bring the copy out. So that's how it works, right? So you have the genes on the DNA in the nucleus, but that gene can't leave the nucleus. So then a copy is made of that gene. That's the mRNA. That copy then can leave the nucleus, which it does. mRNAs leave the nucleus. And now that mRNA is the template, is the recipe. And then the recipe is made, and then you have the product. Chromosomes. So chromosomes are structures. They're actually just DNA molecules, but they're DNA molecules that are highly coiled. They're highly coiled and folded over and folded over, and that's so that it can fit inside of cells, and that's what chromosomes are. Now, chromosomes can be either linear, like our chromosomes are, or they can be circular, like our mitochondrial, the chloroplast, and the prokaryote chromosomes are. So bacteria have circular DNA. Our mitochondria have circular DNA. So genes, as I've been explaining, are segments of DNA that have the instructions or encode instructions for certain products. So usually there are proteins. And then a genome is just all of the genetic information in a cell. It's all of the information, genetic information of an organism. It doesn't even have to be information in a cell. It's all of the information of an organism, all the genetic information. So again, the genetic dogma or the genetic code is then this set of rules that determines how information is passed. So again, information is always from DNA to mRNA, to be more specific, to then protein. Okay, information always starts off with DNA, goes to an mRNA, and then you have the formation of a protein. Just some more terminology: genotype and phenotype. So the genome, as we said, is a collection of all the genes an organism has. So genotype is then the specific genetic makeup of an organism. In other words, what are the genes that an organism has? Or the genotype of a specific characteristic. So, for example, brown eyes. So, brown eyes are coded for by a specific gene. So, the gene that codes for the eye color is the genotype. But then the expression of that genotype is called the phenotype. Okay, so eye color brown, the genes that code for the eye color is the genotype, and then the brown color itself is the phenotype. Okay, so phenotype is the expression of the genes, and genotype is the gene that codes for that expression. So, as I said earlier, bacteria have a single circular chromosome, which is, of course, DNA. Chromosomes are made of DNA. And then the associated proteins, we'll see what they are going forward. On chromosomes, you can have something called repeating sequences of non-coding DNA. So all of the DNA that's on the chromosomes, it turns out, first it was thought that, well, all of these genes that are there, they probably code for something. But it turns out that in that book of recipes, which is the genome, not every recipe produces something. So when you are a gene that doesn't have a product that we know of, right? It doesn't mean it doesn't exist, that we know of yet. It is often just called a non-coding DNA. Okay? So you can have repeated sequences of non-coding DNA in between coding DNA. So coding DNA can be flanked by non-coding DNA, or it could be, a certain number of non-coding DNA segments before it, right? So you have all these permutations of non-coding DNA and coding DNA. So this is just showing a chromosome that's been treated to not be so supercoiled. So this is a one circular chromosome that is that belongs to this bacteria that you see here, this bacillus. So the flow of genetic information will always be from parent cell to daughter cells. The parent cell is going to go through binary fission or mitosis, and now you'll have two daughter cells, and the DNA that was in the parent cell is then going to be the same as in the daughter cells. Of course, there can be problems along this pathway, and then you may not have daughter cells that are exactly like the parent DNA. But for the most part, gene transfers or genetic information transformation or transfer occurs from parent cell to daughter cells. This is called vertical gene transfer. So vertical gene transfer occurs when a cell goes through division, and now you have two cells, and those two cells are going to be the same genetically as the parent cell, and that is vertical transformation or transfer. You can also have a situation like this, where you have the DNA, right? So DNA again is the blueprint of all the coding for the proteins a cell makes. DNA can be obtained via the vertical transfer that we saw, or it can be obtained from another cell from the same generation. What that means is two parent cells can share or can exchange DNA material. Okay, so what you're seeing here, so your DNA can be obtained from another cell in the same generation or from a parent cell during cell division. So the cell division is vertical transfer. This getting DNA from another cell in the same generation is called horizontal transfer, right? DNA then can be expressed within the cell or transferred to another cell through replication. So here's our DNA. Here is the circular DNA within this bacteria. There's a mRNA made from a section of that chromosome or that DNA, and then you are now producing the product. Okay? You can have information, genetic information can be transferred horizontally between cells of the same generation. So here's the DNA. Let me bring it in closer. Here's the nascent or the native DNA for this bacteria, but it's obtained this piece of DNA from another parent cell. Okay, so it's just obtained DNA information, or so that's horizontal transfer, and vertical transfer is just this cell or either one, doesn't matter, went through, they're exactly alike, went through mitosis, and you now have two daughter cells. So this is vertical transfer. This is horizontal transfer. Okay, I'm going to stop here. I'm going to stop here because from this point on, I'm going to stop recording and I'll post this recording.